right now the pathway is inactive because there is a GDP attached to the RAS, and the first step that happens is that the ligand, EGF, comes into the binding site, and this causes the two monochers to come together and become a dimer, and it acts as a dimerase.
This monomer phosphorylates this tyrosines, and it will extract 6 ATP. So each of the ATPs has phosphate, and the phosphate will go and attach to each side of the tyrosines, and the same thing on the other side, and then it will produce 6 ATP.
Okay, so in this step, the adapter protein GBR2 is binding to the phosphorylated intracellular membranes of the RTKs. So it goes here, and this is the adapter protein, and it binds here. And then later, the RAS GEF protein binds to this adapter protein, so it comes here. And by binding to this adapter protein, there's a RAS guanine nucleotide exchange factor, RAS GEF. So this just helps it bind, and then it's activated. And once it's activated, the GDP is released, and it is replaced with GTP. And this GTP is bound to the RAS, and it's activating the RAS here.
The RAS binds to the RAF and activates it. Then the activated RAF phosphorylates the MEK, and it adds a phosphate.
The activated MEK protein then phosphorylates ERK. It's part of the transduction. And then the activated ERK phosphorylates many downstream proteins, such as the transcription factors C, MYC, and it phosphorylates it. And then the CMYC enters the nucleus and activates genes. The CMYC transcription factor enters the nucleus, and this activates cell cycle genes, which continue throughout until eventually the cell response is